Hi everyone, my name is Xiaohui Zhu. Here, I will present our lab's recent work, a simple method to determine the apparent quantum yield of system photobleaching. The apparent quantum yield, AQY, is jargon to describe the efficiency of photochemical reaction. If you are not familiar with this word, you could regard it as the reaction rate per unit of light is absorbed. This research has been published on environmental science and technology. If some parts of my presentation interest you or show a connection with your research, welcome to download it from my ResearchGate page to find more detailed information. As a member of our AquaWatch community, I think you are already familiar with CDOM, the Chromophoric dissolved organic matter. CDOM has strong absorption in UV and blue band. It makes CDOM one of the major controls of water body color. CDOM helps us to monitor the water quality with remote sensing and provides information of the aquatic carbon pool. After CDOM absorbs light, the energy in the photon might break down its molecules, making it lose its color. This process is called system photobleaching. Quantifying system photobleaching in the global ocean is essential in solving a big issue in our community: the retrieval of dissolved organic carbon, DOC, in the open ocean. As we know, CDOM is a good proxy for dissolved organic carbon in the coastal area. This is because these areas are heavily influenced by chromophoric carbon input. Thus, the ratio of CDOM to DOC is consistent with its length source and is relatively stable in this area. However, the relationship between DOC and CDOM becomes a mystery in the open ocean. The biogeneration, biodegradation, and photobleaching of organic carbon are all essential regulators of this ratio. If you want to evaluate the largest oceanic organic carbon pool from satellite, it's necessary to model CDOM photobleaching quantitatively. Even though quantifying CDOM photobleaching is meaningful, it has not been done precisely due to CDOM photobleaching's dual spectral dependency. The excitation of photobleaching is the photon absorbed by CDOM, and the response of photobleaching is the absorption loss of CDOM. Both the excitation and response of this process are spectral. As a result, the AQY of CDOM photobleaching should be presented in a two-dimensional way. For example, a matrix. The complexity to determine a matrix is much higher than to compute the traditional AQY curve. To solve this problem, machine learning could help us to explore the latent relation matrix between the two spectra and optimize the relation with iterations. In our method, the filtered water samples were incubated under the light from a lab or solar simulator. A series of cut-off filters were covered on these samples. They provide different irradiance treatment. A water bath circulator fixes water temperature at 5, 20, or 35 degrees. This is to investigate temperature impacts on CDOM photobleaching. The CDOM incubation was stopped at 12, 24, 48, and 96 hours. Sample absorption change were measured at this time. The amount of photon absorbed by CDOM in this sample cell was computed with direct irradiance measurement. With this computed excitation and response of CDOM photobleaching, CDOM photobleaching AQY matrix was firstly revealed by the partial least square regression, and then was optimized with the gradient descent method. The effectiveness and accuracy of this method has been validated with independent cross-validation and self-validation. The photobleaching AQY determined by our method gives us a new tool to analyze CDOM's properties. Here is a AQY determined from our 0 to 12 hour photobleaching experiment conducted at 20 degrees. We can find that high energetic UV photons are more efficient than less energetic visible photons in activating CDOM photobleaching. Besides, the peak in AQ1 matrix was observed at the excitation response pair of 310 to 180 nanometer. Moreover, there is a diagonal pattern in the AQ1 matrix. It indicates that photobleaching is the most efficient state near the wavelength of exposure. Here is a 20-degree experiment AQY in different time range. The spectral features of the matrix were also dramatically changed in the whole solar exposure history. The diagonal pattern dominates the early stages photobleaching AQY, for example, in the 0 to 12 hours AQY. This pattern gradually gives way to a directional feature. This feature centered at the UVB excitation band under 305 nanometer and extending across the entire response wavelength range. This phenomenon could be well explained by the charge transfer absorption of CDOM. The intrinsic absorption wavelength is decided by the energy gap between the electron's ground state and the excited state. However, when there are electron donator and acceptor pairs coupled with the electron transition, the energy gap decreases due to the energy release in charge transfer. As a result, charge transfer pair redshifts CDOM's intrinsic absorption wavelength. In photobleaching progress, the charge transfer pairs are exhausted gradually. As a result, the long wavelength absorptions get lost in the early stage. This is shown as a disappear of the diagonal pattern. And the later stage photobleaching is more dominated by the intrinsic absorption. It's the reason for the corner pattern. This figure shows how the average value of the AQ1 matrix change with experimental water temperature and the total photobleaching time. We can find that the photobleaching efficiency decreases with the total photobleaching time. This is easy to understand. The CDOM that is liable to photobleaching gets depleted firstly, and then the turn goes to semi-liable, semi-refractory, and so on. The temperature seems to have little effect on the spectral features of the matrix, but its effects on the magnitude are obvious. The photobleaching efficiency increased gradually when the temperature increased. At first, the magnetic gap due to temperature could be as large as 56% between 5 and 35 degrees. This gap decreased with the total photobleaching time and ends at 28% for the 0 to 96 hour experiment. Why is it changed a lot? After absorption, the electron transited to the excited stage and might generate a radical. In high temperature water, the radical moves faster and has a high potential to lead to degradation. Besides, this phenomenon indicates a potential. The ocean warming might be accelerating the global storm photobleaching. This means there will be high dose of harmful UV for marine life in the surface ocean. I will check the potential in my future work. Thank you for listening.